Welcome to the Health Headliner podcast by Tudu Digital. Our content is informational only and not medical advice. Please consult your provider. Now, let's dive into today's headlines.
Imagine a world where a single shot could protect you from almost any venomous snake out there. Sounds like science fiction, right?
It really does.
Well, get ready, because we're diving into some truly groundbreaking research that's making this seem less like fiction and more like a potential reality.
Yeah, there's a new study out looking at a 3-part kind of cocktail
Right.
that's shown really broad protection against the venoms of 19 different medically significant elapid snakes.
And Elapids, that's your cobras, mambas, taipans, kraits, coral snakes, the really dangerous ones often.
Exactly. Some of the most venomous snakes we know.
So our mission today for you listening is to understand how this universal anti-venom idea works, why it could be such a massive game changer for snakebite treatment, and what hurdles are still left.
And we'll try to do that without getting too bogged down in the super technical jargon. We're drawing on a detailed scientific paper, but also some health news reporting to keep it accessible.
The goal is you leave feeling informed and maybe genuinely excited about this scientific step forward.
Yeah, it's pretty remarkable stuff, the potential here to really transform how we deal with snake bites, which are still a huge global health crisis.
Okay, so let's start there. The problem this research tackles, snakebite envenoming. It's actually classified as a neglected tropical disease.
That's right. And that means it causes a massive number of deaths and disabilities every single year, particularly in tropical regions, but doesn't always get the spotlight or the research funding it desperately needs.
And a huge part of the problem with treating it now is that current antivenoms are usually very specific, right? They only work against one or maybe a few types of snakes.
Exactly. Which leads to a cascade of problems when someone actually gets bitten.
Like what?
Well, first off, identifying the snake. That can be incredibly difficult, maybe impossible, especially if it happened quickly or at night.
Right, you might not get a good look.
Precisely. And second, even if you do know the species, the correct antivenom might just not be available where the bite happened, especially in, rural or remote clinics.
And I read that even within the same species, the venom can vary, like geographically.
That's another critical point, yes. Venom composition can change based on location, diet, age. So an antivenom developed using snakes from one region might not work as well against snakes of the same species from another region.
Oh. Yeah.
And then there are issues with the antivenoms themselves. Traditionally, they're made by immunizing animals, like horses or sheep, and harvesting their antibodies.
And that can cause reactions in people.
It can. Things like anaphylaxis, which is a severe allergic reaction, or serum sickness, a delayed reaction, they're not ideal.
Sounds like a logistical headache, too, just keeping track of all these different types.
Oh, absolutely. Stockpiling, distributing, ensuring they're stored correctly, getting the right one to the right place at the right time. It's a massive challenge for a health system.
Okay, so that sets the stage. The researchers saw these problems and aimed for something. Well, pretty ambitious.
Extremely ambitious. They wanted to develop a broad-spectrum antivenom. One single treatment that could work against a really wide range of these elapid snakes.
Because elapids are responsible for a lot of the really severe neurotoxic bites globally.
That's right. They focused on creating a cocktail, a mixture of agents that could neutralize the key dangerous components commonly found in these elapid venoms.
And they used a specific method. You mentioned step-by-step.
Yeah, they called it an iterative addition method. Basically, they started by targeting the Elapidae family generally, knowing that their venoms often rely heavily on two main types of toxins. These things called three-finger toxin and enzymes called phospholipase A2s.
So three-finger toxins and phospholipase A2s, those were the main targets. Where did they find the ingredients for this cocktail? This is the really wild part of the story, isn't it?
It really is fascinating. They identified a human donor, someone with an extraordinary personal history. This individual had spent 18 years intentionally immunizing themselves against a huge variety of snake venoms.
Intentionally? You mean like injecting small amounts?
Apparently so. The paper mentions over 800 immunizations involving venoms from mambas, cobras, rattlesnakes. So those are vipers, taipans, kraits.
Yeah.
Just an incredibly diverse range.
18 years. Wow. I mean, that's dedication, but also that person's immune system must be incredible.
Exactly. The researchers saw this as a unique opportunity. They analyzed blood samples from this donor.
And what did they find?
Compared to people who'd never been exposed to venom-naive individuals, this donor had way higher levels of antibodies, specifically against key neurotoxins. Things like the long neurotoxins from black mamba, Cape cobra, coastal taipan, and common krait.
So clear evidence of broad immunity developing. Were the antibodies themselves different? Better somehow?
Yes, that's another key finding. The donor's antibody repertoire showed a much higher rate of something called somatic hypermutation.
Somatic hypermutation. Okay, what's that in simple terms?
Think of it as the immune system's editing process. When antibodies are made, this process introduces tiny changes, mutations, trying to find versions that bind better and stronger to the target, in this case, the venom toxins.
So it's like an accelerated evolution for antibodies.
That's a good way to put it. And this process was even more pronounced in the antibodies that were specific to venom components, and highest of all in antibodies targeting these long neurotoxin relatives. This person's immune system had basically generated super-optimized defenses.
Incredible. So this long-term diverse exposure really trained their immune system. How did the researchers actually pull out the specific antibodies they needed for the cocktail?
They use a technique called phage display. It's a powerful method that lets you screen enormous numbers of antibodies relatively quickly. They screened antibodies produced by the donor's memory B cells.
Those are the cells that remember past infections or exposures.
Exactly. They remember the threats, and using phage display, they fished out the antibodies that were best at binding to and neutralizing the key venom toxins.
And they found two main ones.
Two key broadly neutralizing antibodies, yes. One they called LNX-D09.
LNX for long neurotoxin.
Precisely. LNX-D09 was highly effective against those long neurotoxins, and the second one was SNX-B03.
SNX for short neurotoxin.
You got it. SNX-B03 was broadly reactive against the short neurotoxins.
Okay, so long and short neurotoxins. Are they both types of those three-finger toxins you mentioned earlier?
Yes, they're both members of that family. They have similar core structures, but slight differences, particularly in how they interact with their target in our bodies.
Which is?
Which is a receptor on our nerve and muscle cells called the nicotinic acetylcholine receptor. That's what these neurotoxins attack to cause paralysis.
Okay, got it. So LNX-D09 for long neurotoxins, SNX-B03 for short ones. Let's take LNX-D09 first. How does it actually work? How does it stop the toxin?
This is where structural biology comes in, looking at the exact 3D shapes. They saw the crystal structure of LNX-D09 bound to a long neurotoxin.
So they could see exactly how they fit together.
Exactly. And they found the antibody latches onto a very specific conserved region on the toxin. Conserved meaning it's basically the same across many different long neurotoxins from different snakes.
And why is that region important?
Because it's the exact same spot that the toxin uses to bind to that nicotinic acetylcholine receptor, its target.
Ah, so the antibody gets there first and blocks the toxin.
Precisely. It physically obstructs the toxin's binding site. It's almost like the antibody mimics part of the receptor itself to fool the toxin and block it. A really elegant mechanism.
That makes sense. And because that site is conserved, the antibody works against toxins from lots of different snakes.
That's the key. It hits a common vulnerability.
Did they test if this antibody worked in, actual living systems?
They did in mice.
Yeah.
LNX-D09 alone provided complete protection against lethal doses of purified long neurotoxins from various elapids.
Okay, against the pure toxin. What about the whole venom, which is more complex?
Even more impressively, yes. It gave complete protection against lethal doses of whole venom for multiple cobra species, black mamba, and even the king cobra.
Wow. Even when given after the venom, like a treatment.
Yes. Even in post-exposure scenarios in the mouse models. This strongly suggested that these long neurotoxins are really the primary killer components in those particular venoms.
That's huge. But what about the other toxin class you mentioned, the phospholipase A2s?
Right. They knew LNX-D09 wouldn't handle those, so they brought in another component, a small molecule drug called Varespladib.
And Varespladib specifically targets
phospholipase A2 enzymes. It's an inhibitor. It blocks their activity. These enzymes cause different kinds of damage, like tissue destruction and sometimes also affecting nerve function.
So did adding Varespladib help?
It did, especially against venoms where phospholipase A2 is known to be a major player. Think venoms like the tiger snake or the taipan. Varespladib on its own offered some protection against those.
And together with the antibody?
Often, yes. They found that the combination of LNX-D09 and Varespladib frequently provided complete protection against species like tiger snakes and taipans, where neither agent alone was fully sufficient.
So it highlights that sometimes you need a multi-pronged attack.
Definitely. Different venoms have different key weapons, so sometimes you need different countermeasures.
Okay, so that covers the long neurotoxins and the phospholipase A2s. What about the second antibody, SNX-B03, for the short neurotoxins?
Similar story, mechanistically. They also determined the structure of SNX-B03 bound to a short neurotoxin.
And it also binds to a critical spot.
Yes, it also targets a conserved site on the short neurotoxin that's crucial for its binding to the nicotinic acetylcholine receptor. Again, it works by blocking the toxin from reaching its target.
Okay, so we have LNX-D09 for long neurotoxins, SNX-B03 for short neurotoxins, and Varespladib for phospholipase A2s, the full three-part cocktail. How did that perform overall?
This was the big test. They challenged mice with venoms from 19 different Elapid species, all considered medically important by the World Health Organization.
19 species? How did the cocktail do?
The results were pretty striking. The three-component mix gave complete protection against 13 out of the 19 species.
13 out of 19? That's really impressive.
It is. That included snakes like the banded krait, the eastern coral snake from North America, the mulga snake from Australia, a diverse group.
What about the other 6, the ones it didn't fully protect against?
For the remaining 6 species, the cocktail still significantly extended the survival time compared to untreated controls. It didn't offer complete protection, but it clearly had a substantial effect.
Okay. Still beneficial, even if not perfect for everything.
And there was a fascinating finding with the common krait venom. For that particular venom, none of the 3 components, LNX-D09 SNX-B03, or Varespladib, offered complete protection on their own, just partial.
Right.
But when they use all 3 together,
Complete protection.
complete protection,
Yeah.
a real demonstration of synergy, where the combination is much more powerful than the sum of its parts.
That's really cool. And geographically, these 19 snakes, they covered a wide area.
Oh, yes. They represented species from North America, Africa, Asia, Australia, Oceania. And they also spanned a good portion of the evolutionary tree of the Elapid family.
Which suggests...
It suggests that this cocktail, or the principles behind it, might have the potential to cover a very large number, maybe even most, of the over 300 species within the Elapid family. That's the hope.
Okay, let's zoom out then. We've gone through the science, which is fascinating. What are the big picture implications here? What could this mean, for actual snakebite victims?
Well, the headline potential is obviously a universal antivenom,
Mm-hmm.
at least for Elapid snakes. That alone would be revolutionary.
Just thinking about not needing to identify the snake.
Exactly. That critical, often impossible first step, gone. You just administer the broad-spectrum treatment. That saves time, reduces errors, and could make treatment possible in situations where it currently isn't.
And potentially safer, too. You mentioned issues with animal-derived antivenoms.
Right. These antibodies, LNX-D09 and SNX-B03, are based on human antibody sequences. The idea is that fully human or humanized recombinant antibodies would be much less likely to cause those dangerous allergic reactions like anaphylaxis or serum sickness.
So, better safety profile. And maybe easier to use.
Potentially, yes. Human antibodies generally have a longer half-life in the body than animal antibody fragments used in some traditional antivenoms.
Meaning they stick around longer.
Yes, which could mean fewer doses are needed to maintain effective levels, reducing the treatment burden. And researchers might even be able to engineer them to last longer still.
And from a health system perspective, just having one product instead of dozens.
Huge simplification. Think about stockpiling, distribution, cold chain storage, training health workers. Having one go-to product for any Elapid bite would streamline everything immensely, especially crucial for resource-limited settings.
And it could help overcome that venom variation problem you mentioned earlier.
That's another key advantage. By targeting conserved regions on the major toxins, a broad-spectrum approach inherently provides better protection against the natural variation that exists in venom composition, both between species and within species from different locations.
It really does sound incredibly promising. But, science is rarely that simple. We need to be realistic. What are the limitations or the caveats we need to keep in mind here?
Absolutely. It's crucial to acknowledge the limitations at this stage. Firstly, the venom dosage in the mouse studies. They use specific, lethal doses.
But a real snakebite might inject more.
Exactly. A real bite could involve a much larger venom dose. And at higher doses, maybe other minor toxins in the venom that aren't neutralized by this cocktail could start to have significant effects. That needs further investigation.
And as always, mice aren't humans.
The classic caveat. Results in mice are a vital first step, but they don't always perfectly predict what will happen in humans. We'll need studies in larger animal models and eventually carefully conducted human clinical trials to confirm safety and efficacy.
Also, this is all focused on elapids, right? Cobras, mambas, kraits.
Correct. A huge step for that family. But this cocktail wouldn't be expected to work against vipers, for instance, rattlesnakes, copperheads, gaboon vipers. They have very different venom compositions, so we still need separate broad-spectrum solutions for other snake families.
And even within the elapids tested, it wasn't 100% protection for all 19 species.
No, there were those 6 species with only partial protection, extended survival. That suggests that for those snakes, either other toxins are playing a bigger role than anticipated, or perhaps antibodies with even higher affinity, tighter binding might be needed.
And you mentioned the short neurotoxin antibody SNX-B03 wasn't quite as potent.
Yeah, the data showed it had a somewhat lower binding affinity compared to LNX-D09. That might contribute to some of the partial protection observed. Fine-tuning or finding even better antibodies could be necessary.
What about the drug component, Varespladib? Any issues there?
The main one mentioned is its relatively short half-life. It gets cleared from the body fairly quickly, which meant they sometimes needed repeated doses in the experiments. For a practical antivenom, especially one used in remote settings, a longer-acting phospholipase A2 inhibitor would certainly be preferable.
Okay, so definitely still challenges to overcome and questions to answer. It's a proof of concept, but not quite a finished product yet.
Precisely. A major breakthrough, but more work is needed.
So, given both the potential and the limitations, what's the general feeling among experts looking at this research?
There seems to be a lot of excitement. Generally, experts really highlight how this study showcases the power of the human immune system, especially when it's been unusually stimulated, like in this donor. It shows it can be an incredible source for finding broadly effective therapies.
Almost like mining the immune system for solutions.
In a way, yes. And the fact that this donor had such extensive, diverse exposure seems key to generating these broadly neutralizing antibodies.
What about the structural work? Understanding how the antibodies work.
That's seen as critically important. Those insights into how LNX-D09 and SNX-B03 bind to those conserved spots on the toxins. That's not just cool science, it provides a roadmap. It helps guide future efforts to design even better antibodies, or maybe even small molecule drugs that mimic them.
Knowing the target precisely makes drug design much easier.
Exactly. And the overall approach, identifying the key toxin classes first, then developing specific neutralizers for each. That iterative strategy is viewed as a really logical and systematic way to tackle the sheer complexity of snake venom.
A potential template for other venoms too, perhaps.
Possibly. And there was that point about LNX-D09 being remarkably similar in its binding site to another antibody discovered completely independently.
Right, you mentioned that 95 MAT5.
Yes, which came from a synthetic library. The fact that 2 totally different discovery methods converged on the same neutralization strategy. Experts see that as strong evidence that this really is a key vulnerability, maybe even the optimal target site, on these long neurotoxins.
Like evolution and lab design arrived at the same answer.
Kind of, yeah. It suggests targeting these functionally critical conserved sites, the parts the toxin absolutely needs to work, is a very promising strategy. Maybe not just for snake venoms, but for other toxin families or even pathogens.
Fascinating. Well, this has been a really illuminating deep dive. We've journeyed from the problem of snakebite through this remarkable story of a unique human donor to the clever science of antibody discovery and cocktail design.
And seen this really compelling proof of principle for a broad-spectrum Elapid antivenom. It's a testament to innovative thinking tackling a neglected but deadly serious global health issue.
It really is. So, as we wrap up, maybe a final thought for you, our listeners, to consider. Where else could this type of approach lead? We focused on snakebite, but could similar strategies finding broadly neutralizing antibodies, perhaps from highly exposed individuals or targeting conserved functional sites, could that work for other types of envenomation? Maybe spider bites or scorpion stings?
Or even beyond toxins. Could targeting conserved regions be a strategy against rapidly evolving viruses, hitting the parts they can't easily change?
It really makes you think about the untapped potential still hiding within the human immune system, doesn't it? And how we might harness that incredible adaptability to solve other major health challenges. What other neglected diseases out there might just be waiting for this kind of innovative approach?
It definitely broadens the perspective beyond just this one, albeit very important, study. Lots to think about.
Thanks for tuning in to the Health Headliner podcast by To Digital. Like and subscribe for more health news and breakthroughs. Stay healthy and informed. See you next time.